Okay, hi guys. I know there are a lot of questions about how to analyze the crystal violet data, so I'm going to go over that quickly with you guys in this video. Hopefully that will help you process it a little bit easier. So in the crystal violet growth analysis, we've basically taken our samples and stopped them at different time points and stained them with the crystal violet stain. After the experiment is over, we remove the stain, we destain them, we remove the excess stain by washing them, and then we solubilize the remaining stain, which we assume is mostly taken up by actual living cells that were still attached to the plate at the time we ended the experiment. And then we quantify that stain using spectrophotometric analysis. So we are looking at the amount of blue that's in the solution. The more blue, the more cells are there. That's our understanding, or that's our assumption. So we expect to see more and more blue in a typical setting as the cells replicate and divide and grow over time. So the data that is provided to you is for 72 hours, so 3 days of treatment. The first thing you want to do is collect that data and copy it into an Excel sheet. So we're going to do that right here. Now this data contains triplicates of each of our treatments. So you have 3 controls, C1, 2, and 3, 3 Taxols, 3 mulberries, and 3 saffron. So those will serve as your replicates for the experiment. And then you have the absorption after the stain has been solubilized and quantified at each of those time points for each of the sample. Now, if you just look at the numbers for 0 hour, you will notice that most of them are around this 1.2 point, but there are some variability, right? There is some variability. Like this one is 1, even here's one that's 2.17. So there is a little bit of difference in the starting confluence, and we want to make sure to take that into account as we run our analysis. So what we're going to do is the first thing, and in the assignment, it asks you to make a separate graph for each or separate table organizing each of your samples. You can do that following those instructions, or you can also just pretty much copy and paste this exact table right underneath and convert all the numbers at the same time to the relative growth. Now, when we're doing the relative growth, we basically want to convert uh, we want to use our 0 hour time point as our baseline. So I want to say, well, this is my starting number of cells or starting absorbance. I want to see how the absorbance change over time with the treatments base, compared to this baseline, the 0 hour. So we're going to make our 0 hour um, into a 1 by dividing it with itself, and then we're going to divide everyone else in that column by that first number as well to see, relatively speaking, compared to 0, if the cells grew or died and how much they grew or died. So when we do that, we can do that using a formula. So I'm going to write equal sign, and then I'm going to make it a little bit bigger if I can. I won't do it right now. Okay, 1 second. So maybe it's a little bit easier to see. Okay, right there. So equal sign, and I'm going to take my first number and divide it by itself. Now, if I wanted to make sure that it was this cell that I wanted to divide everything by, all the numbers and all the replicates, then I would want to put a dollar sign both before the column and before the row. However, I actually want it to shift columns as I move to the next sample because I want to take the 0 hour of each replicate to be the baseline for that particular sample. So I do not want to put a dollar sign next to the column, but I do want to put a dollar sign next to my row because it's row 4 that contains the 0 hour data point. So I will put that, and then it gives me a 1. Now, just to make sure that I did this correctly, I can, take it all the way down, and all the 0 hour should say 1, and then I can drag it down and I will get my other numbers as well. Now, that is one way to do it. We could have also done it where we took an average of the absorbance at 0 hours for control and then use that number for all the samples. That's another way to do it as well. Either one is fine, but we are going to stick with this one for this exercise. So once we have done that, now we can pretty much, we have all the data and we can go ahead and get our standard deviations and our averages for the graph. So I'm going to copy it, the time points, and I'm going to write the treatments along the top. So it's going to be my control, Taxol, right, mulberry, and saffron. And then I'm going to calculate the averages. So average, it's going to say, what do you want to average? I want to average these 3 numbers. Oops, control C. Now the 3 controls for 0. And then I'm going to go ahead and change the formulas for the other treatments. So it's the correct cells, T1, 2, and 3. And then for mulberry, in my case, it's going to be I, starting at I, and saffron's going to start at L. Okay, so once I have my averages for the 0 hour, I can pull it down and it will give me all the other numbers. And now that's going to give me my basic relative growth average numbers from the 3 replicates for each sample. I'm going to make another graph, not graph, another table just like this, but this time I'm going to have standard deviations. So again, I'm going to create my formulas for the first row and then copy them down. So I'm going to take my standard deviations of these 3 numbers, and then that's the one that I want to copy. So standard deviation, I'm going to change the cells for my Taxol, then my mulberry, and then my saffron. And then I can drag them down and get my basic that. Now to create my graph, what I will need are my actual numbers up here. That were my averages that I calculated. You're going to select all that data set, including the titles on the top and the side, so the time points as well as the actual treatments. You're going to go to Insert, Scatter Plot, and right now it is plotting. So if you look at this, it is plotting the incorrect way, where it's plotting time as individual points when you want your treatments to be the points. So you can go to data, click on this, and chart design, go to chart design and say switch rows to column, and right there, this is the basic graph that you want, where you have basically each one of your treatments as lines and your time as x-axis, your average of the relative growth as the y-axis. And now you can, format it to meet the needs and do all the corrections you need to do, all the changes you need to make to that. Now, finally, what you need to do is But this just gives you the basic idea of what that graph is going to look like at that time. Now, the last thing that you need to do is to do statistics, is to run your statistics. And there are multiple things that you need to do for that. The first thing that you need to do is run a two-way ANOVA, since this time you have two separate independent variables. One is your treatment itself, what type of treatment they were given, and the second is the time point. So we have over time, so we have different time points, and we have different treatments. So it's a two independent variable analysis, so it's going to be a two-factor ANOVA. Again, for this, you are going to need, under your data, you will need the data analysis tool. If you do not have that, you would need to go into Files, Options, Add-ins, and then go to Excel add-in at the bottom, click Go, and then select Analysis Toolpack and click OK. I already have it, so I'm just going to leave it there. Now, what I need to do is organize my data in a way that Excel can recognize it as two independent variables, and it knows which replicates are there, and it can analyze them properly. Now, for Excel function to run this two-way ANOVA correctly, the replicates have to be in rows. Right now, your replicates are in columns. They are, going along the top. We need to flip this. So to do that, I'm going to select the data that I provided you, right? So I'm going to take this entire table. I'm going to copy it. I'm going to come down here, and I am going to paste special and transpose. By transposing, you've essentially just rotated your numbers so that now your time points are along the top and your treatments are along the side in rows. And so that's where your replicates are. Now you're ready to run your two-factor ANOVA. You would click on the Data Analysis tab. You are going to get your two-factor ANOVA with replication. That's the one you're going to run. You are then going to click on this up arrow and select this entire table that you just made. Everything, including the treatments and the top, which was your time points. You are then going to come in here. It's going to ask you how many rows per sample do you have. So you're going to say 3 because we have 3 samples per treatment. And then for output range, I'm just going to tell it to place the tables right next to the original table and click OK. So now when I run the two-factor ANOVA with replication, I get several tables, one for each treatment. So it's just calling it C1 arbitrarily because the first well was labeled C1, but this is your control, Taxol, Mulberry, and then Saffron. And it's going to give their averages and standard deviation, or variances rather. But then at the bottom, you will have your two-factor ANOVA. And that's going to contain your significance. It's going to contain your degrees of freedom, your F value, the F critical, and then your 3 p-values. The first p-value it will have is sample, which basically looks at each one of your treatments, so samples that were on your in the rows. So it's comparing the control, Taxol, Mulberry, and Saffron. Your columns contain your time points. So the p-value here, it's looking at just the effect of time on each one of your treatments by themselves. It's not looking at cross differences between them. It's just looking at each one separately. And then finally, the third p-value that you have is interaction. And that looks at how the samples behaved over time in response to the treatments against each other. So it showing significance here would mean that different treatments responded over time differently. So some grew while others died. If all of them grew or all of them died regardless of the treatment, then this would not be significant. The fact that this is showing significance indicates that there is at least one sample, one treatment in your data set that behaved differently over time than the others. So you're going to be using those to interpret the data along with the t-test that you're going to run. You will run the same 6 t-test for each time point, not the 0 hours, but afterwards you're going to run for 24, 48, and 72. So you're going to have 18 different t-tests that you will run, and you'll use those to interpret the results. So hope this helps to clarify some confusion and gives you a little bit more insight into how it is that we analyze this type of data set a little bit better. Okay.